Hello everyone, welcome to MES e-learning channel. So we will continue with our genome editing series of lecture. In this particular lecture, we will talk about TALENs. What is TALENs? Transcription, Activator-Like Effector, Nucleases. So here, as we all know, again this is an engineered protein which is a combination of TAL, which is what is TAL? Transcription Activator-Like Effector molecules along with the fork1 endonuclease, the same nucleus that we had seen in case of your zinc finger nucleases also. So first, let us understand about the DNA binding specificity of TALs. So this is also going to have, these TALs are tandem repeats of 33 to 35 amino acids, and these are called as modules. So if you see in the diagram, the different colors which you are able to see, the blue, green, pink, so each color is actually identified as one module, and every module is going to recognize a single DNA base pair. So that is the reason why with the help of these tails, we can program the targeting. So like whatever our target sequence is there, we have to incorporate those particular modules together and make a combination such that the desired DNA could be identified and recognized, so that it will help the nuclease, that is your fork1 nucleus, to have the double-stranded break. So it's a very flexible design. It can be modulated depending upon our need. So the creation of TALENs with the wide range of DNA binding specificities is the important characteristic of this particular methodology. Moving ahead. So this is the fork1 endonuclease that we can see the 3D structure also. So once the TAL binding domain has bound to the DNA, we have this fork1 endonuclease which are fused with these TALs will cut the DNA and double-stranded breaks would be introduced. So it requires, fork1 requires dimerization for the double-stranded breaks to be introduced, and here you can see that two sets of TALENs, so a TALEN pair we call it. So two sets of TALENs are going to bind to the opposite the DNA strands, so that highly specific DNA cleavage could be achieved with the help of these particular engineered proteins. So DNA binding, first the DNA will bind with the help, or the TALs will bind to the target sequence which is there with high specificity. Then the cleavage would be done with the help of the fork1 endonuclease, and then the cellular repair either by NHEJ or your HDR, that is your homology directed repair, would be activated to modify the target DNA. So this mechanism will remain the same for all the genome editing tools. So advantages, as we have seen that it is customizable. So the TALENs can be designed for different types of your DNA sequences. What we have to just do is change the combination of the modules. So that can be achieved very easily, and then improved efficiency. So it is demonstrated higher efficiency in targeting the DNA, targeting and creating the DNA modifications as compared to the zinc finger nucleases. And multiplex targeting, that means simultaneously the different types of targets can be modified, okay, and edit several genome loci at a single experiment. So this is also one important characteristic of TALENs, and so we can use these TALENs for variety of different applications for the genome editing tool. Again, limitations are there for TALENs also. Sometimes off targets can be there, so it can potentially bind and cleave unintended genomic sequences also, so that unwanted modifications are there. Delivery challenges, same as zinc finger nucleases, is a problem in case of your in vivo applications. So regulatory hurdles, we have to follow, we have to pass all the regulatory barriers and the concerns which are there for safety and ethics. That what is the future developments that we can go for? So first we need to improve the specificity, so that there would be reduction in the off targets, optimize the delivery which is required, so we have to develop efficient delivery methods so that we can use this particular tool for in vivo modification. Regulatory oversight is going to be there for all the genome editing tools so that we can address all the problems, all the concerns which are there, and combinatorial approaches. So as we are having different types of your genome editing tools, if there is a way in which we can combine them together, it may give us, or it may have an enhanced capability for the genome editing. So these are the different future developments we are looking ahead. And with this, I hope you have understood the concept of talents. Thank you so much.